Okay, so we're looking at a picture here that shows us the 4 steps of the crossbridge cycle. So during the crossbridge cycle, remember from your studying that this word crossbridge is just a fancy word for myosin attaching to actin and making the actin move. So there's 4 things that happen, and when we talk about what's happening in them, the things that you want to keep track of in every stage, we want to know what's going on with the shape of myosin. So is it standing straight up? Is it kind of bent over? Is it really unattached? Those kind of things. So first thing we want to know is what's going on with myosin. The second thing that we want to know is we want to know what it's attached to. So myosin is really the star of the crossbridge cycle because this is what's going to pull on actin and make actin move. So when we start here, step number 1, the name of step number 1 is crossbridge formation. So this is not a trick question here. When we talk about what's happening in the stage of crossbridge formation, what might we say is happening here?
Contraction.
Well,
the
so
myosin is moving.
Myosin doesn't move yet. Myosin is actually going to move here. We'll add that to our next step here. So myosin bends right here. When myosin bends, that causes movement. So the movement part actually happens here in step 2 in what we call the power stroke. So think about the power stroke as what powers your movements, what makes things happen. So myosin bends in the power stroke. That's causing movement because it's pulling actin. Actin's what actually slides past.
Okay,
Over here in
this
the first
case,
step. Oh, go ahead.
sorry. Okay, the first one, then the myosin is attached to the action.
Exactly. Yep. So the only thing that happens in the crossbridge formation step is myosin and actin attach. And remember from our notes, we called this fancy word here crossbridge. Crossbridge is just a fancy word for when myosin and actin attach to each other. So the fact that this stage is called crossbridge formation means that I'm forming a crossbridge. I'm attaching myosin and actin to each other. That's the only thing that happens here in crossbridge formation. Now, for me to make this happen, for myosin to be able to attach to actin, it has to be in this position you see right here, where see how myosin is pointing straight up. So we have a fancy name for the position that myosin had to be in. So myosin was in they call it the cocked position. The cocked position is I'm ready to go. I'm standing straight up and I'm ready to attach. So for us to be able to have a crossbridge form for myosin and actin to attach to each other, myosin needed to be standing up. If myosin was standing up, it can reach up and it can attach to actin, crossbridge formation. Now, after it attaches to actin, what happens is the myosin realizes it doesn't need ADP and this phosphate anymore. I kind of like to joke with students that originally myosin is attached, we can see down here in our last step, it's attached to this ADP and this phosphate, and it thinks that it's happy because it's got these things that it's attached to. But suddenly when it has the option to attach to actin, it's like it's made a new best friend, and these friends aren't important anymore. So in this next stage, in the process of the power stroke, myosin actually spits out this ADP and this phosphate, so it lets go of those things. When it lets go of those things, it actually changes its shape from being standing straight up to being bent over now. So during the power stroke, 2 things happen. The first thing that happens is it spits out the ADP and the phosphate. And then the second thing that happens is it bends over, which makes the actin slide with it. So we'll make a note here for ourselves. ADP is released. When I release that ADP, that's going to cause my actin to move, where this is where the movement stage is. When I get to this point, though, when myosin has bent over and it's pulled actin with it, now it's kind of stuck. What happens next is a molecule of ATP comes along and attaches in the same place that we used to be attached to ADP. ATP comes and attaches to myosin. When ATP attaches to myosin, it's the same thing that happened up earlier when it attached to actin. Myosin attaches to ATP and says, oh, I don't care about actin anymore. I've got ATP. I'm happy now. So in this third stage here, let's scroll down a little bit, crossbridge detachment. In the crossbridge detachment stage, myosin detaches from actin, meaning it's not holding on to each other anymore. So in step 3, myosin and actin let go. They split up. We're done attaching them to each other. But the other important thing in that stage, this is where ATP attaches to myosin. We have to have ATP attaching to myosin to make myosin and actin let go. So otherwise, myosin and actin would stay attached forever. And that's actually what happens, if you remember from studying lesson number 9, that's what happens in rigor mortis, when the body gets really stiff. Myosin and actin would stay attached forever because I have no ATP to split them up. So if ATP comes and attaches, myosin is going to let go of actin. But now myosin, if you can see my picture here, is bent over. It's in the wrong shape. So what myosin is actually going to do in this last step, it's called cocking the myosin head, or in one of the videos that you watched, it's called the reactivation of the myosin head. When myosin is bent over and it's in this wrong shape, it's not going to be able to attach to actin ever again. So what it does is it takes this ATP, which we talked about, why does a cell like ATP? What is ATP for a cell?
Energy.
Yeah, that's its energy. So myosin is attached to this energy molecule. Myosin says, hey, if I steal the energy that's stored in ATP, if I break up ATP and turn it into these things, so I split it apart, I can go back into the cocked position, the standing up position. That would allow me to attach to actin again. So this last step here, called cocking the myosin head or reactivation of the myosin head, we break up ATP and get its energy. So ATP is broken. It turns into that ADP stuff that we saw, that we started with at the beginning, and we can see it right here. So I break down ATP and make ADP instead because I chopped it up. And then the other thing that happens is myosin stands back up. Myosin is now upright again, and myosin will be able to attach to actin. So once it's back in the cocked position, that's where we started right here, right? So if I'm back upright in the cocked position, now I can attach to actin again. Now we can start this cycle over where I'll bend and spit out these low energy molecules, get more energy, start the process over. So those are the 4 steps of the crossbridge cycle. So let me pull up the homework assignment here and we'll chat through some of the questions that you had to see if we can figure out which of these 4 stages was being described in each question. So one of the questions that we had was said that this was the stage where ATP attached to myosin. Which stage does ATP attach to myosin?
ATP, it's the crossbridge.
Yeah, the detachment stage, right?
Yes.
This one down here. ATP attaches to myosin. That happens in crossbridge detachment. That's going to make myosin and actin split from each other when ATP attaches. We also had a question that asked about when the energy that ATP had is released and myosin changes its shape. Which stage did I use the energy in ATP to change the state of myosin? To change its shape, excuse me.
It's the power stroke.
So the power stroke doesn't have ATP, actually. The power stroke has
ADP. Yes, ADP.
Yeah, so which step did we say that myosin stood back up? It got into the position where it's ready to attach to another actin.
last
Yeah,
one.
that last one down here. Yep, cocking the myosin head. This last step right here, this might be a good note to make for yourself, actually. The only step of the process of muscle contraction where we actually use energy, like where I chop up a molecule of ATP to make energy, is actually this last step right here. So cocking the myosin head, taking the myosin protein from in the wrong shape back up into the right shape, that's the only step in this whole process that I actually use energy for. So cocking the myosin head is when I use the energy of ATP to change the shape of myosin. It's a good point that you mentioned, though, that myosin does change its shape in this state too. Do you remember we talked about why myosin changes its shape when we're doing the power stroke? What did it do to make it change its shape?
Because they bend over.
Yeah, so we're trying to figure out what was different between this right here, so this is at the end of Crossbridge formation, and between this right here. What was it that myosin did that helped it to bend over? Do you remember what happened?
ADP is released.
Exactly. Yep. So what causes myosin to change its shape over here to actually do this power stroke where we make power, where we contract, is it spits out ADP. So when I spit out ADP, like we put right here, that's going to cause myosin to pull on actin and make actin slide. When we're talking about taking myosin from the bent over, unable to do anything shape into the cocked position where it's upright, I'm going to chop up the ATP to make that happen. So one of the questions you actually had on your homework assignment was about that power stroke stage. It said, which stage does myosin let go of the low energy ATP molecule that it was attached to? That's what we just talked about, right? This is
Yes.
stage, the power stroke, is when myosin spits out that ADP. There's no energy in that molecule. There's no reason for it to keep holding onto it. When I spit that out, that's going to cause me to change my shape. And that allows me to do the power stroke. Let's see if I can think of some other questions while we're staring at the Crossbridge here. Let's see. Well, here, let's do an application question. We talked a little bit about this, but when I'm talking about rigor mortis, when the body gets stiff, when you die, which of these stages did we say can't happen when we have rigor mortis? Which stage gets blocked? Do you remember?
In rigor mortis, let me see. Okay. I know that the myosin and actin stay together.
Yep, they're stuck together.
They stuck together, yes.
And when you die, you stop making something. What is the chemical molecule that you stop making when you die? My hint is that the mitochondria make it for you.
ATP?
ATP. Yep. When you die, you stop making ATP. When ATP attaches to myosin, that causes this crossbridge detachment stage. So when you die, what happens is you get stuck right here. We get stuck at the end of the power stroke where we've spit out our ADP and our phosphate. Actin has slid, it's moved across. Now myosin is in this bent over shape and it's stuck attached to actin. It can't do anything right here. Normally, when you're alive, when your muscle cells have mitochondria that are working, this ATP molecule would come and attach to myosin and say, hey, myosin, time to let go. We got to let actin move back to where it wants to go. But if I have no ATP, myosin and actin get stuck together. So in rigor mortis, when your muscles get really stiff, what happens is I can't move on to step 3. I'm stuck at the end here of the power stroke. I can't move on because I have no ATP to help me move on. So if I had a question that asked which stage ATP attached to myosin, it attaches to myosin in this stage right here, Crossbridge Detachment. And I think we talked about this one when I break apart ATP to change the shape of myosin. Now we're over here to cocking the myosin head. So a reminder for us, the things we said to watch in each of these stages of the crossbridge cycle. The first thing that I told you to watch is the shape of myosin. So is myosin upright? Is it bent over? Is it attached to actin, or is it not attached to actin? That's the first thing we want to know for each of these steps. The other thing that we want to know is what else it's attached to. So is it attached to ADP? Is it attached to ATP? Or when we're up here in our power stroke state up here, we're just attached to actin. We're actually not attached to ADP or the phosphate there. Both of those spit out. Does that make more sense now that we chatted through it a little bit?
Yes, now makes more sense. I was confused.
Mm-hmm.
I was reading and
go ahead.
No, I was reading and rereading this, and I said, "Gosh, I cannot like figure out." It's like I think it was too confused. But now that you explained to me, yes, now it's more clear.
Perfect. And I know we have a pretty good video that talks about the process of the crossbridge cycle too. So as you continue studying it for the next exam, definitely check out that video, because instead of just seeing the pictures where myosin is in one shape, right, and it's all blocky like this, it'll show you the process of myosin changing its shape and then letting go and all that kind of stuff. So check out those videos, because I think that would be helpful for you as well.
Okay, thank you.
Let me look back over your attempt as well. I think that was, that's probably the big one. We talked about the types of muscle tissue. So just review, it looks like we had a simple question on here, the parts of the sarcomere. We just want to make sure we know where each part of the sarcomere is, and that ties into lab actually. So make sure you can find things on those lab models and on our pictures where the different parts are, like the Z disc and the I band, those things that show us the arrangement of actin and myosin. I saw you sneak in, Amy. Welcome. Did you have questions about number 9 or number 10? Which one were you working on?
Well, number 9, can you hear me?
Yes, I can hear you.
Okay, great. Have you already talked about the Sarin and Botox questions?
We talked a little bit about them in office hours last week, but we could talk about them again if you'd like to.
Yes, I just can't seem to figure out exactly, how to answer those correctly.
Okay, let me see. I have, we'll start with this picture right here. Ooh, zoom out, if it'll let me. Oh, okay. So this picture here is something that you have, see, there we go. Okay, that I made for you, that I told you it kind of outlines the whole process of how muscles contract. So from the beginning, where a neuron sends a message down through the muscle fibers membrane, all the way down to what we just talked about with the myosin and the actin. Okay. When we're talking about sarin gas or when we're talking about Botox, both of those are going to give me trouble at this stage up here. So something that we talked about in office hours last week, the process of muscle contraction, overall, big picture, we call it excitation, contraction, coupling. That's just a big fancy word for saying that the excitation part of its name means that I have a message from a neuron, and the contraction part obviously is muscle contraction. contraction is muscles. So I have to have a message from a neuron to cause contraction to happen. Now, in normal scenarios, we'll zoom in, it'll be a little fuzzy here, but in normal scenarios, my neuron is going to release this little chemical that we see, the little green dots we see inside our neuron, called acetylcholine, ACH.
Yes.
So
Okay.
normal situation, neuron, which you're learning about in lesson number 10, neuron spits out acetylcholine, these little dots down here, and these little acetylcholines go down, they attach to a chemically gated channel and cause my muscle fibers membrane to change its charge. So it goes from being negative on the inside to freaking out and being positive on the inside.
Okay.
That's the way that I get a message from a neuron into a muscle cell.
Okay.
So we had two things that we talked about were problems with this. The first thing that we talked about that was a problem with this is something called Botox. So Botox, the thing that bacteria make, right? Botulinum toxin.
Right.
When we talk about Botox, what do you remember about what Botox does? Do you remember anything about it?
Yeah, I wrote down that it blocks the acetylcholine, you know, and all the, it's like it stops the chemical signal from reaching muscle.
Exactly.
So it reduces contractions.
Yes, absolutely. So I like the way you said it. It blocks the chemical signal. I like that. What happens with Botox is this toxin gets inside the end of my neuron here. And when it gets inside, see how I've got these little bubbles? They're called vesicles that store those chemicals. If this neuron is trying to talk to this muscle fiber, it's got to empty out the contents of this bubble into the space down here. For me to be able to take it from this bubble to spit it out and make it get into the space where it can go talk to the muscle cell, I actually have to have, and here I'll draw a couple for us here. I have to have special little proteins down here in the bottom part of my neuron's membrane. So there's all these little proteins that are found all along the end part of my neuron. These little proteins act kind of like a dock for this bubble. So if this bubble comes down here and attaches to these proteins, then it's going to spit out the chemicals like I see all of these other bubbles doing. So what Botox does is it gets inside this neuron, and Botox goes through and it actually chops up all of these little docking stations right here.
Oh.
So it goes in and it chops up the proteins. Now I have these little bubbles that are storing acetylcholine for me, except they're floating around forever inside the neuron because there's no place for them to dock so they can spit out their acetylcholine.
Okay.
So you're absolutely right. When we said that Botox, it blocks the chemical signal, and then we can add to that then that it blocks contraction.
Okay.
The problem with Botox is we don't contract at all.
Okay.
And
so vesicles can't exit.
Exactly. Yep. They can't exit, so the chemical messages are stuck inside the neuron.
Okay.
And this is why when somebody gets Botox, why like the gag is right, that like they can't smile anymore or they can't talk anymore. Like in the movies, they always do it bad. Yeah. So we basically paralyze the muscles. So they can't contract anymore because they're not getting any messages. All of these messages are stuck inside. That's the problem with Botox. Let me find my picture to help us talk about how Sarin works. I'm going to pull up actually the whole page that has.
Yeah, on both of these, I thought I understood, after the videos and reading the pages. But I just really couldn't seem to figure out the questions.
So for Sarin, we'll use this picture down here. So this picture down here, let me color code it to make it look a little bit more like what we were looking at here. This is my neuron. So it was kind of yellow or orange, right, in the previous picture. So I've got my neuron up here. So my neuron has acetylcholine. There it is inside a little vesicle here. when your neurons spit out acetylcholine, like we see happening here, here it's going down and it's attaching to those channels that we just talked about, the message going down to. Now, when your muscle cell gets this acetylcholine message, we only want it to respond to it once. We only want your muscle cell down here to contract when the neuron says it's time to contract. So the neuron spits out a message, muscle cell contracts, and then the muscle should stop. It should be done contracting. The way that we make sure that happens is this enzyme acetylcholine esterase. Okay. And that's going to be our big thing here for sarin gas is this guy right here, acetylcholine esterase. Do you happen to remember what acetylcholine esterase does, what its function is?
I think it causes the acetylcholine to detach. I'm trying to think of the pictures. Let me look. I guess it breaks down acetylcholine. That's what I said before. So that, and then I guess the acetylcholine can be, it's either broken down and recycled, or sometimes can it be used again? Can it re-enter the neuron for future use? You know, the presynaptic.
Yes. Okay. So for my muscle cell to respond to acetylcholine, it has to be in this big form that we see here. So the acetate group and the choline group, they're attached to each other, acetylcholine. Right. What acetylcholine esterase does is it actually chops up the acetylcholine and turns it into an acetate and turns it into a choline. Okay. When it's split up like this, there's a couple of things that means. The first thing, like you mentioned, is I can recycle it a little bit. So I can take this choline that I chopped up, that neuron, the presynaptic neuron grabs it, brings it back inside, and then turns it back into new acetylcholine that I can spit out again. So that's the first thing. That's what I do with the choline. But the reason that I'm going to chop it into acetate and into choline is because my receptors, like I see right here, they're only activated or they only open up when acetylcholine attaches to them, not when acetate attaches to them, not when choline attaches to them. The only way I can open up this channel is I have to have this particular form of the chemical. It has to be acetylcholine. Okay. If I have any other form of the chemical, that's not going to open up my channel anymore, which basically means I'm not going to keep contracting. Okay. Right. And go ahead.
Oh, I was going to say, so it's like specificity, right? That's the word.
Yeah, that's a great way to think about it. So I think the word they use in a general biology class is that enzyme specificity. Right. So we have, even though my picture only shows one, we can add a couple more of those channels, right? That are on the membrane. These are the things that normally we'd open with our acetylcholine, like we see over here. Well, if I just have acetate or I just have choline, this binding site right here is specific for acetylcholine. And neither of these are acetylcholine. Okay. So these will not open up for me.
Okay. Now, does the sarin attach to those protein receptors? Is that what happens?
It does not attach to the protein receptors, not to these guys. Okay. Sarin actually attaches right here to our acetylcholine esterase. Ah. Okay. So we'll put a little note here. Sarin attaches to acetylcholine esterase. There's my abbreviation there. Sarin comes in, attaches to acetylcholine esterase, and when sarin attaches to acetylcholine esterase, this enzyme can't do its job anymore. So we said the job of this enzyme is to break down acetylcholine. When sarin gas attaches to the enzyme, I can't break down acetylcholine anymore. So what might happen if I can't break down acetylcholine? Or we could flip it on, maybe think about another way too, with this question here that you had in your notes. Why did we say it's a good thing that I break down acetylcholine?
To stop contraction? You don't have
contraction, right? So that's the goal of acetylcholine, is to stop contraction. Well, sarin is stopping acetylcholine esterase from doing its job, from stopping contraction. So if I block this enzyme right here, now this acetylcholine that I used to chop up, that used to not send a message anymore, if I don't chop it up anymore, that acetylcholine will be a message I receive right here, and it'll cause a muscle contraction. And it'll detach from here and float around. Hey, look, there's another place that I can attach to. And I'll attach here and cause a muscle contraction. And then I'll let go right here. And hey, look, I found another place to attach, and I cause a muscle contraction. The problem with deactivating acetylcholine esterase is your neuron sent one message to contract 5 minutes ago, except acetylcholine isn't getting chopped up. And I just keep hearing the same message over and over and over again because it goes and it finds new receptors to attach to. So when sarin gas is released, what it specifically does is it blocks the activity of acetylcholine esterase. What ends up happening then is the acetylcholine that you released, you keep responding to it repeatedly, which can lead to things like muscle spasms, which can lead to things, ultimately, things like your muscles running out of energy completely, and then they just stop contracting. So like when we were talking about sarin gas with, here, I'll pull this up here, sarin gas in Syria, what usually makes sarin gas fatal, if it's going to be fatal, is respiratory failure. Because muscles like your diaphragm, they got a message to contract, and they just keep contracting and keep contracting until they just run completely out of energy and they can't contract anymore. Does that make more sense, Amy? I may have lost Amy.
How about now?
Okay, I can hear you again.
Okay. Yeah, it just, my mic switched off, but.
Got it. So does that make more sense to you?
Yes, I believe so.
So, well, and so here, let's go down. I think there is, let me do the next page here.
A lumberjack.
So here is where, I guess we're on, looks like I'm on page 13 now.
Okay, I'm there.
Yeah, so we just talked about actually a lot of these questions right here. So we just talked about what acetylcholine esterase does. You told me on the previous slide that the job of acetylcholine esterase is to break down acetylcholine. That's its job. What might we summarize and say the job of acetylcholine is? I think you already kind of told me this. What does acetylcholine do?
A decomposition of acetylcholine.
Oh, well, sorry. So now I'm talking about the acetylcholine itself. That's the.
Oh, it okay, I can't remember. It causes the muscular contraction. It attaches to the protein receptor and triggers, I guess, the action potential, or the contraction of the muscle. Oops, I can't hear you now.
Oh, I think we lost her.
Yeah, you can't hear her either.
Oh, sorry about that. My internet died.
No problem.
So it wasn't just.
Whoops.
Oh, I think she's there.
Maria, are you still there?
Yes, Amy, I am here.
Yes, I'm here. Okay. She'll be back.
Sorry, my internet is not happy over here. Okay.
Oh, no problem.
So we said that sarin gas, it impacts acetylcholine esterase, right? It stops it from doing its job. So when we talk about sarin gas, what happens to my acetylcholine? Do I end up having more of it or less of it being heard by my muscle cell? Does it look like I have a lot or a little?
More.
Yeah, it looks like I have a lot, right? So how does sarin gas impact the level? It looks like there's more acetylcholine. I put looks like because my neuron just releases the normal amount, but I can't chop it up. So we're going to say it looks like there's a lot of acetylcholine. So my question over here that said what might happen if I had too much acetylcholine? Well, you can read about what happens with sarin gas because sarin gas is a problem with too much acetylcholine. And so that ties in then when we're talking about Botox, about what's going on with Botox. With Botox, we don't release any acetylcholine, right? No acetylcholine is released. And I need acetylcholine to have muscle contraction. So this is why, excuse me, after Botox, a person can't contract their muscles because we're not releasing any acetylcholine. So to answer our question here, what happens if I don't have enough acetylcholine? Well, just look at what happens with Botox, because with Botox, there's no acetylcholine being released. And that leads us to not be able to contract.
Yeah. Makes sense.
Well, and what you can, I was just going to say what you can do too is now that we've talked about these two things as well, you can always go back to your homework assignments and look at the particular answers and see why you might have gotten something wrong.
Yeah.
Maybe now with understanding it a little bit better, maybe that'll help us to kind of read into those answers a little bit more.
Yeah, I expect so.
Maria, were you going to say something?
Okay. How about when a person who always is like injecting Botox? Okay, of course, the muscle is not going to contract and the skin is going to be tight. But if they keep doing it like every time, can it lead to any other problem?
So the reason they have to keep getting Botox is because your body actually goes through and clears it out. So a person gets Botox, I think it lasts, I used to know this, I don't think it lasts more than like 3 months. By the end of 3 months, your neurons have gotten rid of all of those botulinum toxins and they've actually rebuilt those new proteins that would help us to dock those bubbles. So getting Botox done regularly doesn't lead to problems. But what can happen is if your Botox is injected incorrectly, so maybe it's supposed to go to the facial muscles, but someone accidentally injects it into the bloodstream, then it ends up going throughout the body and it acts the same no matter where it is. So whether it's going to the muscles of the face or whether it's going to the muscles of your heart or the muscles of your blood vessels, that's why you have to be really careful with injecting Botox because it always does the same thing wherever it goes. It makes our muscles not contract. So inject it very carefully. Make sure it's just going into those muscles right there by the surface of the skin, for example. But that then will lead to those muscles not contracting.
Okay, I got it.
Yep, we got it.
Perfect.
Okay.
Any other last minute questions for today?
No, thank you.
No, I have for lesson 10, but tomorrow. Okay.
Okay. That sounds great. I will plan to see you tomorrow for lesson 10.
That sounds good.
All right. You ladies have a good night.
You too. Thank you.
Bye.
Mm-hmm.
Okay. Bye.
Bye.